Thanks, Ekaterina, for the introduction. I'm Shan Huang, a postdoc from Huang's lab at UCLA. I'm glad to share with you our recent exciting work about how sex information of a social partner is encoded or represented in the prefrontal cortex and how those sex representations shape animals' behavior. Social behavior is essential for animals' health, survival, and reproduction. Many types of social behaviors are evolutionarily conserved, so they exhibit similar features across species. And such behaviors include social affiliation, social dominance, mating and parenting, etc. Many types of social behaviors have a preference for partners of a specific sex. For example, dominance behavior is usually observed between animals of the same sex, while mating behavior is usually observed in animals of opposite sex. So you can see that animals display very different types of behavior, which is dependent on the sex of the other animal. Therefore, being able to recognize sex of other animal is critical for animal to make behavioral decisions. So how is sex information processed in a rodent brain? Those chemosensory cues, such as odorants and pheromones released by other individuals, are mainly processed in main olfactory system or nasal system. And the two systems converge onto a region called medial amygdala, where sex-specific neural response start to show. And then they further diverge into different hypothalamus centers to trigger sex-specific behaviors, such as aggression, mating, and parenting. However, little is known about how sex is encoded beyond those subcortical regions. Cortical circuits are thought to be more plastic and adjustable than subcortical regions, since they can quickly accommodate outside changes and allows for adaptive behavioral decisions, such as dominance and sexual behavior. In addition, it has been shown by our lab and other groups that medial prefrontal cortex, or mPFC, is involved in sex-dependent behaviors. mPFC is also known for its essential role in social cognition and decision-making. Therefore, we would like to investigate how sex is encoded in this area. The main approach we applied is calcium imaging using Miniscope, which is a small version of fluorescence microscope with a super lightweight and has single-cell resolution. The small animals can wear this scope while they are freely behaving, so we can monitor their neural activity during certain type of behaviors. So this shows an example of Miniscope recording. We usually can image hundreds of neurons in a single field of view. Through downstream video processing pipeline, we can extract the activity trace of each neuron and align them with behavioral events to analyze how a particular neuron is responsive to a certain behavior. The first question we wanted to ask is whether mPFC neuron encode male and female specific information. So during Miniscope imaging, we presented each subject mouse with multiple novel males, novel females, and a toy mouse in a random order. The subject mouse can freely explore each novel intruder for about 20 seconds, and after the whole session was done, we extract the single cell activities and classify cells into categories of female responsive, male responsive, toy responsive, or unresponsive to any stimulus. Here shows some example cells that have a selective response to either male or female investigation. Next, we quantify the number of male or female responsive cells. In female animals, we observed an equal fraction of neurons that were excited by a male or a female social partner. And interestingly, male animals have a larger fraction of neurons that excited by female investigation. This finding is quite intriguing as it suggests two things. Number one, the encoding of sex information in mPFC differed between male and females. Number two, male animals have a stronger representation of female information than the male information. Next, we want to take a look at how sex is represented at the population level, which should give us a more stable and reliable response than individual neurons. We first simplified the data using principal component analysis, or PCA, which is good at transforming a large set of variables into a smaller one that still contains most of the information. The response of hundreds of neurons were mapped onto this three-dimensional space defined by the top three components. Take this animal as an example. It showed a clear separation of population response during male versus female interaction, suggesting that the population activity can distinguish sex information. In addition, we found that male animals showed a higher amplitude of population response during female investigation compared to male investigation, which is consistent with the previous finding that I showed that male animals have a larger fraction of neuron encoding female information. In the following analysis, we focused on male animal to gain deeper insight into this asymmetric encoding of sex information. So why males have a stronger encoding of females, and whether the stronger encoding of female has anything to do with their behaviors. We looked back behavioral experimental videos and wanted to see whether animals have a preference for partners with a specific sex. We measured the time that animals spend with each social partner and calculate the sex preference score, with positive number meaning a preference for interacting with males and the negative number meaning a preference for females. We observed a group level bias toward interacting with females, raising the possibility that the female encoding bias may be linked to this female behavior bias in male animals. Although they show a female preference in average, a substantial fraction of males show a preference for interacting with male animal. So where this individual variability of sex preference come from? And we hypothesize that individual sex preference may be due to its own asymmetric encoding in the mPFC. To test this hypothesis, we first grouped individual animals into female preferring and male preferring groups based on their preference score. We found that the female preferring animals showed a much stronger response during interaction with female social partners than with male partners. In striking contrast, male preferring animals showed a stronger population response during interaction with male animal. This result suggests that the relative activity of male versus female trigger response can predict sex preference behavior. And this will explain the individual variability in sex preference. To test whether there is a causal link between cortical sex representation and sex preference behavior, we would like to manipulate the activity of sex encoding cells and see whether that can influence animals' behavior choice of interacting with male or females. However, it has been difficult to specifically manipulate those sex encoding cells as they typically do not have a simple biomarker, such that an activity-dependent labeling strategy is necessary in this scenario, which can specifically label and manipulate those neurons based on their natural response to a different sex. The particular construct we used is driven by a synthetic promoter called ECR. Here, we adopt the ECR activity-dependent labeling coupled with optogenetic stimulation by injecting two viruses, an activity-dependent Cre recombinase and the Cre-dependent channelrhodopsin. Neurons that were naturally activated by a male or female social stimulus would express channelrhodopsin down the road, so we can use optogenetics to stimulate those native sex ensembles. And this expression process can be switched on by tamoxifen injection. This diagram shows how we carry out the experiments. Each subject mouse was given 1 hour to freely interact with novel male or female in their home cage. After 1 hour social exposure is done, we systematically injected tamoxifen to activate Cre recombinase. A few weeks later, we test the subject mice in a two-chamber assay where subject mouse can freely choose to interact with either male or female partners. We shine blue light in the middle of the experiments to stimulate the sex encoding ensembles and see whether that makes any changes on sex preference behavior. The control group, okay, got the time. The control group was not given any social stimulus and they were otherwise handled in the same way as the experimental group. We used optogenetics to stimulate neurons that were naturally activated by female cues. Stimulation of female activated cells in the middle of 10 minutes resulted in an acute change in sex preference toward female animals. And in contrast, stimulation of male activated neurons dramatically reversed an overall female preference to male preference. And you can see that after stimulation, they go back to female preference. And this suggests that sex preference behavior is modulated by the activity of native neural representation of male and of female cues. So let's take a summary. I have shown that different sets of neurons in the mPFC respond selectively to male and female cues, and they elicit different population patterns. And the population response amplitude can predict their sex preference. And then when we stimulate those native sex responsive cells using optogenetics, they can directly modulate animal sex preference behavior. And sometimes they can even reverse the original preference to the other sex. This demonstrates a causal role for activity of social representations in shaping social behaviors. And this finding demonstrated for the first time that sex information can be represented in your cortical region. So this will allow for more adaptive behavior decision to accommodate outside and internal changes. Thanks, thanks everyone, and welcome questions.
Okay, thank you very much, Shan. I think it was very fascinating study, especially the part that you were able to actually manipulate the social specific cells and change their sex preference or change their behavior towards the other sex. So that was very interesting. And I think we have time for just a quick question. So if I don't see any questions in Q&A box, but please ask questions. So the question that I have is whether do you have any idea what type of cells those are within the prefrontal cortex that are responsive to the sex-related cues?
Good question. So we did differentiate the chemokines two, like the excitatory cells and inhibitory neurons, but we didn't find a difference between them. So both excitatory and inhibitory cells, they can encode sex information. And there's not a number difference, but it seems that the sex information is more enriched in the inhibitory neurons. As if you normalize them into the same number of neurons, then inhibitory neurons contain more information about sex. But based on other literature or other groups of single cell sequencing, there seems not a difference in molecular composition of the cells. So it's hard to tell what type of subtype of neuron will encode sex. It seems like it's well distributed across different types of cells.
I see.
Great question.
Very interesting. Okay, thank you very much. I think that's all the time that we have for the questions, and you can reach out to Shan on Twitter if you have further questions, or you can ask on Q&A panel. So now we're going to move on to our